Well, thank you so much. I'm so delighted to be here and to see you attend my lecture. It's a terrific honor. I don't take it lightly at all. It's just fabulous, especially considering how many outstanding colleagues I have here. I was truly surprised when I was informed of this and did not expect it at all, but I'm delighted to receive it and delighted to tell you about some of the work that my group has been involved in over the years. I'd first like to say a little bit about Alfred Holmes White, for whom the professorship is named. So I chose him primarily because of his long service to the University of Michigan, and in particular the Department of Chemical Engineering. He was there at the very beginning in 1898 and served the department for 40 years. For 28 years as chairman of the department. I thought I served a long time for 8 years. And he brought the department to the first ranks of excellence in the country. He also, his name was used as well by Donald Katz, who was a pioneer and one of the outstanding figures in chemical engineering. Donald Katz was the first A.H. White professor. So what I'd like to talk about today is a shaping of materials. And the shaping of materials goes back to the dawn of human civilization, and it's been critical technology ever since the beginning. But it's only been in the last 100 years or so that we've begun to shape materials that are somewhat similar, at least, to the materials that are used in biology. A very good example is the silk threads that are spun by a spider. To this day, as I understand it, silk threads, spider threads, are tougher than any known material on a per weight basis, and yet they're processed at room temperature with water as a solvent. So it's truly a green processing operation. And we are beginning to mimic some aspects of that as we see processing large quantities of polymeric materials for everyday applications. But there are many problems that are encountered and defects that occur in the processing of polymeric materials. Films can tear, there can be defects on the surfaces such as the shark skin, or even worse, instabilities that occur in shaping these materials, and we'd like to understand these so we can control them and process better. Because these are complicated materials, one strategy is to look first at simpler materials for which the instabilities and flow properties are somewhat better understood. And we can look around in nature and see many instabilities that occur in simple fluids like air or water, or in the kitchen where you see instabilities that occur. One of the best studied of these is in the flow between two cylinders. So you have an inner cylinder rotating and fluid contained between it and an outer cylinder. And if you spin the inner cylinder fast enough, an instability develops that's illustrated here. And it's caused by centrifugal force, which tries to throw the fluid from the inner cylinder to the outer cylinder, but it has to push out of the way the fluid that's already there. And that creates a pattern like this. And there's a whole stack of these so-called roll cells that appear. And if you spin the inner cylinder faster and faster, you get additional instabilities that eventually lead to full-fledged turbulence. So polymeric materials are way too viscous for these kinds of inertial or centrifugal instabilities. For example, here's an especially viscous polymeric material. It's a toy, but it's called silly putty, but it has the properties of a lot of these polymers. So what can cause instabilities in processing polymers? Well, my colleagues and I, several years ago, back at Bell Labs, discovered that this rotational flow between two cylinders leads to an instability even with polymers that are very viscous. So here's what we found with polymeric fluids. And we know that this is not a centrifugal instability. It's not due to inertia, because the fluid is way too viscous. And moreover, if you rotate the outer cylinder instead of the inner cylinder, you still get this instability, which you would never get in the case of the centrifugal instability with simple liquids. So we want to understand how this forms and how ubiquitous it is with other polymers. So this was done with my colleagues Susan Muller and Eric Schockfey. So in explaining this, I'd like to take a bit of a tour. First, I'd like to show another instability that's closely related to the instability between cylinders, and that's if you have two disks, two parallel disks, one of them rotating. You can also generate a cascade of instabilities that are shown here. These images were produced by my graduate student, Bruce Schamberg, leading to very irregular behavior that you see here that's come to be called elastic turbulence. So how do these kinds of instabilities occur? So I'd like to take a tour of 4 principles that are used for controlling and shaping polymers and other materials along the way in explaining this. And these are Brownian motion, elasticity, self-assembly, and then we'll talk about some futuristic work in self-propulsion. So we'll start with Brownian motion. Brownian motion was discovered by Robert Brown. He was looking through a microscope at little particles that came from pollen grains. And he saw they perpetually danced around like this. These are images that Mike Solomon took in our department. Brown got very excited because he thought he had discovered something called the vital force. And at the time, it was believed that living things had a special force called the vital force that would animate them. And he thought that's what he discovered. So he's quite disappointed to learn that dead things do exactly the same thing as long as they're small enough and suspended in a liquid. They will move around like this. So that may be why he looks so grumpy here when he discovered this. So the true explanation for this behavior actually was first articulated 2,000 years earlier by this guy, Lucretius. He was one of the early believers in the existence of atoms. And his insight was so great that he said that these atoms would be in restless movement. They would be driven on in ceaseless and varied motion. And in fact, that's exactly the case. That motion is caused by collisions with solvent molecules battering that little bead from all sides, pushing it around in a random motion. And Einstein was the first one to analyze this and show how to interpret it and to explain it as being the result of atoms and molecules. And at the time of Einstein, actually, the existence of atoms was still in dispute. There were holdout physicists like Ernst Mach who opposed this notion. And in fact, Einstein's analysis became one of the most definitive proofs of the existence of atoms at the time. Okay, so it turns out then Brownian motion is ubiquitous. It's really nothing other than waste heat, which will exist in all fluids. And it's taken advantage of, especially by the cell, because Brownian motion is the primary means by which molecules in the cell find each other and bind and carry out their day-to-day activities. And a particularly good example occurs in the nucleus of the cell, where the human cell has 2 meters of DNA packed in the tiny space of a nucleus, and it has to be packaged in chromosomes just to keep some order. But you can see all these levels of structure that exist down to the individual DNA molecule. And if you digest away the packaging, a single chromosome looks something like this. And if you zoom in on this little rectangular area, here's what you see. And somehow the cell has to read this and find the information that it needs moment to moment in order to continue its operation. So how does it do that? How does it find the information it needs? How does it read the right part of the DNA? Well, it does it by Brownian motion, using proteins that will find the target site by moving about in this jittery fashion. And it's believed that this is aided by the protein first finding the DNA and then moving along the DNA to the target site. So it would be much like waking up in a dark room trying to find your dresser, and you first find the wall and then move along the wall. And so we were one of the groups that tried to actually image this process. Okay, so we weren't the first to do this, but we used a method in which we aligned DNA molecules onto a glass slide, and this was done by graduate student Ji-Hoon Kim. And it was important that the molecules be attached only at the ends to this slide so that the rest of the molecule was free to interact with protein molecules that are brought in by a flow in a perpendicular direction, and then we image them and watch to see how they interact with the DNA. Okay, so here I'll show a video of this, and the DNA in this case is invisible. You can't see it, but you'll see that it's there because when we bring proteins in from the side, they begin to bind, and you see the individual binding events, and there are many places for binding of these proteins on this particular strand of DNA. There's another example here where the protein concentration is less, so it takes longer for proteins to find the DNA, but they eventually do. Now, to look for this process of searching, the Brownian process of searching randomly, we replaced this DNA with a DNA strand that had no binding sites. And when that's done, you see the protein moving back and forth in this characteristic random fashion known as Brownian motion. And we can analyze that motion by looking at a time series like this. You see it going up and down. And if you take the distance traveled in a given interval of time and do many experiments and bin up all the results, you get a curve that should be familiar to many of you. This is called a normal distribution or Gaussian distribution, and it's a characteristic of a random process. The width of this, when you square it, is linear in time, and the slope of that gives you what's called the diffusion coefficient. This turns out to be much slower, much smaller than is the case of the protein moving in bulk solution. So why is it so slow? Well, the reason we believe is because the protein actually has to read the DNA. It's looking for a particular sequence. So in this experiment, the DNA is tethered at one end onto a surface, and then flow stretches it out. Again, you don't see the DNA, but the proteins that are brought in stick to the DNA, and they're pushed along in a kind of irregular, herky-jerky fashion. And we believe this is because the proteins are looking for their binding site. They're looking for a particular base sequence here of 8 or 10 bases. And when it finds something that's close, it's sticky. It tries to stick and then realizes it's not the right spot, and it has to escape from this trap to move further. And so this will greatly slow the motion of this protein as it searches along the DNA. And so we've analyzed that, and we find this sort of herky-jerky motion can be predicted because there are long periods where it's trapped temporarily. And so the diffusion coefficient goes down rapidly, as is predicted, if you make the depth of this rough surface, so-called free energy surface, greater. Okay, finally, we looked at transcription. So the DNA is on a surface, and we can add in what are called nucleotide triphosphates. These are used to build RNA chains. These are the transcripts, which are like photocopies of pieces of the DNA. And those transcripts are imaged here. And this video here is showing you in real time the motion of the protein, which is moving in this direction along the DNA, transcribing, making this chain as it goes. So here it's moving in only one direction. There's no Brownian motion. And the reason it can do this is because these bases, these nucleotide triphosphates, carry little power packs with them. And that energy source is used to grow the transcript chain and push the protein further in one direction so that it can convert the DNA information into the RNA transcript. Okay? We tried to mimic this process and found it to be very hard. So the idea here in this grant that involved a number of researchers, including Jim Baker and his group, was to take a molecule called a dendrimer, which is protein-like in its size and shape and carries positive charges. So it should be able to stick to negatively charged DNA, and we hope to see it move diffusively along the DNA, proving that the interaction was mostly due to charge. But we found it didn't do that, and in fact, in solution, the dendrimer causes the DNA to basically collapse into these little bundles. When we simulate that, we simulate the dendrimer interacting with the DNA, we find that the DNA is wrapped around the dendrimer like string on a spool. And we believe this interferes with its ability to move along the DNA. But this is related to other problems of interest, including what are called histones. These are structures that are part of the chromosomal structure. It's the wrappings that you get in packaging DNA into the chromosome. So what would it take to get an artificial protein to slide along DNA? And looking at actual proteins, you find that this is an example here where the protein is shaped so that the DNA fits like a hand in glove, and the charges on the protein seem to be strategically located in order for the DNA to be able to configure itself. So we hope to be able to learn more about this by designing dendrimers that have specific shapes and charges. Okay, so now I want to move on to elasticity. And to do this, we'll start back with this problem of coating DNA onto a glass slide. So I mentioned that we wanted the DNA to stick only at the ends. And this, we believe, happens because the ends of the DNA have bases inside that are greasy and will stick to a greasy surface or an oil-loving surface. So the surface that we use, the only kind of surface that will work for this, is a surface that doesn't like water, doesn't like the water droplet, but likes oil. So we believe the ends of the DNA stick, but not the middle. And to demonstrate this, my student Ji Hoon deposited a DNA chain on the surface and then cut it with a beam of light. And you'll see that the DNA snaps back to two positions. You can see it repeated here, indicating that the DNA indeed is just stuck at the two ends, leaving the rest of it free for the protein to interact with. You can also see here how elastic the DNA is. It snaps back like a rubber band. And this elasticity is again caused by Brownian motion. Every little piece of the DNA is trying to move in all directions randomly. And so this will lead the DNA to very, very quickly coil up into a blob because of this random motion. So if we look at free DNA, long DNA in solution, it coils up into little blobs, and then these dance around by Brownian motion. But if you put flow on, as Steve Chu and his group did, you can see that the DNA will stretch out. If you turn the flow off, it will collapse again. So this becomes a basis to understand polymer elasticity, and we can begin to get at the problem of how polymers are elastic and how that affects their flow properties. So we've simulated this in very clean flows where the experiments were done at Stanford. And you can see the DNA stretching here. And using Brownian motion and fluid mechanics, we predict the way the molecule will stretch out in the flow. This is called a dumbbell mode of stretching. And it agrees extremely well with what's measured. But there are other modes. I'll show you just one other, which is called the fold mode, where the DNA acts like rope over a pulley. And you should see here that it's brighter on the top half. This simulation shows you in more detail what's happening here. And this is another way that the DNA can unravel. And so by looking at this in great detail, we can understand how long polymer molecules are stretched in a flow, and then we can apply that to other polymers, such as the ones that are used to make products. So an example is here, where a collaborator in Australia is using this device to stretch out drops of polymer into long filaments, the way perhaps a spider would stretch out a liquid into a filament. And you can see how you can stretch it out and it doesn't break. You'd expect it to break up into little droplets. It does not break up into droplets because of the large elastic stresses that develop as the polymer molecules are stretched. And in fact, in this device, you can measure the forces and determine what the stress is as a function of time after you turn on this flow. And you can see as you stretch the filament, these stresses get to be very, very large, especially if you go at high rates. In fact, you can get to 300 times the stress that you have in the fluid without the polymer, even though there's only 0.1% polymer present. So it's a huge effect, and this is the basis for spinning fibers that we use in fabrics, for example. And if you get bored with my talk, you can demonstrate this to yourself by taking a little bit of your saliva, and if you pull it out between your thumb and forefinger, you will see a little filament form, and it's remarkably stable. Okay, if you do it right, which I learned to do in grade school, and I guess I kind of got bored, but maybe it was a precursor to the work I'm doing now. But anyway, this is the basis for the large elasticity that enables processing polymers into sheets, large sheets, and into fibers. And the lines here are the predictions that we made of these forces based on what we learned initially from the DNA molecules and how they are stretched out. Now here's another application of the elasticity. And those of you who work in the kitchen have seen this probably. If you try to beat cake batter, you'll see the batter climbs up the shaft of your egg beater. And that's what's happening here. This is a polymer solution. And this is called rod climbing. The reason this occurs is the polymers are stretched out now, and they wrap partially around this shaft. Of course, they're too short to wrap all the way around, but the polymers are curved, and this curved, stretched shape of the polymer causes it to pull the fluid inward, and it drives the fluid up the shaft. So this is like the opposite of what we see with a centrifugal instability or centrifugal force, where you expect rotation to be throwing the fluid outward. Here that effect is very weak, and what's strong is the elastic force pulling the fluid inward and pumping it upward. So now this is the clue that lets us explain the instability that I showed earlier. That is, if we have fluid in the gap between two cylinders and subject it to flow, the stretched polymers want to push fluid inward. That pushes other fluid out of the way, and it has to cycle around. Now it turns out when we solve the equations for this, the fluid mechanical equations, it's quite a complex flow that's not even steady. It's time periodic with these vortices that appear at the inner surface and actually move across and then repeat themselves in an endless cycle. And this is all caused by these elastic forces in the polymer. And this basic instability shows up in many processing applications of polymers, including the parallel disk flow that I mentioned, and even flow in channels is now suspected to have instabilities related to this phenomenon. Okay, now we can try to use this elastic force for good purpose as well, and this is some work that we're just starting with Sunitha Nagrath, who is developing little microfluidic devices to separate cancer cells from normal cells. And they can be separated based on the different stiffness of the cell. So this is a simulation showing a flexible cell and a stiffer one. And you can see that in this curved geometry, the interaction of the flow and the stiffness of the cell can cause a separation. So this will be useful for detecting cancer cells, and we'd like to be able to understand this process. Okay, now I'm going to go briefly through self-assembly and self-propulsion. So self-assembly is very important in biology. It's only beginning to be utilized in commercial applications of polymers. But one simple way to get a kind of assembly is to make the polymer self-attractive, using charges, for example, and stiff. And then as you turn up the attraction, the polymers will collapse, and they will collapse into different shapes, either these bundles or toruses or globules. And these are reminiscent of what's seen in biological polymers, for example, in DNA or proteins. Another kind of self-assembly occurs with what are called lipids, which self-assemble into these bilayers, which are the membranes that cover every cell. And this can also occur in self-assembly also involves proteins and peptides, which will take on specific orientations in the interior here of a lipid bilayer. And these are very important in transporting ions and information across lipid membranes. Another application of self-assembly with lipids is in what's called lung surfactant. So when you breathe, you have little air sacs that expand and contract, and the work of expanding them is so great that you couldn't breathe without a surfactant that decreases the surface tension and lets these sacs expand. But then when they contract, the lung surfactant has to go somewhere. So in these simulations, during this phase when you're exhaling, we find that the lung surfactant will form these little pouches. They're little folded regions facilitated by peptides that are present in the lung surfactant. And if these are absent, as they are in very premature infants, you can't breathe. So that's known that you need this in order to enable proper breathing. So one of the interesting applications here is to design artificial lung surfactant components that could be of use therapeutically. Finally, I'll mention particles that can self-assemble because they are not homogeneous. And an example is the so-called Janus spheres. This is the god, the two-faced god Janus. And these spheres have an attractive side and a repulsive side. And so they will assemble into different patterns as groups such as the granite group have shown. And also there's very beautiful experiments by Mike Solomon and Sharon Glotzer. Glotzer is doing the simulations of these. In our group, this is work by Daniel Beltran in collaboration with Mike and Sharon. We find that these kinds of particles can self-assemble into lamellar structures that are shown here. And these are like very poor man's proteins. So finally, I'll get to the last topic, which is self-propulsion. And this is something that's quite a hot topic now, to try to make artificial self-propelling materials. So large animals swim by using inertia. So basically, in one way or another, you throw water behind you, propelling yourself forward, using the equal and opposite, for every action there's an equal and opposite reaction. So you use the inertia of the water that you throw behind you to propel yourself forward. But tiny organisms like E. coli are too small to have any significant inertia. So they have to find another way to swim. And the way they do it is they have little corkscrew tails. And they literally screw themselves through water. They rotate these tails, propelling themselves through water. And they have multiple of these tails called flagella. And they can use one of them to steer, like a rudder. And you can see these little guys here from Howard Berg's website, steering themselves around. These are quite fantastic machines, really. This is a video that shows what's really going on. There's a motor here, which looks like an outboard motor, but it's not powered by gasoline, it's powered by protons. And that rotates this shaft around, and it goes around 100 times a second. And then drives this flagellum. And these flagellums spontaneously bundle together. And they do that because of the stiffness and because of the fluid flow that they generate. So we want to understand in detail how this worked. And so my student Nobu Watari developed the first whole body model of an E. coli bacterium that includes the flagella, the body, which is built out of little beads and springs, kind of a tinker toy model that has the mechanics in it, as well as the flexible hook that connects the flagellum to the body. And then there's a torque balance you have to satisfy. Again, Newton's laws at play. And this allows prediction of the swimming and the correct swimming speed. And here you can see the bacterium tumbling and changing its direction. So it changes its direction by counter-rotating one of its flagella. And that causes it to tumble and then take on a somewhat different direction once it starts swimming straight again. We find that the tumbling, the angle through which the bacterium will change its direction is very sensitive to the location of the flagella on the body. So you can get large tumbling angles or very small ones. And we're comparing these now with experiments from Howard Berg's group that also show a very large distribution of angles that you get. So there's some very efficient rotations or changes of direction in the bacteria, and some that are very inefficient. Bacteria actually can smell food, and they can direct their turning so that they rapidly find the food source. Okay, so I've given you a little bit of a snapshot of what goes on in my group, and I wanted to thank those who really did the work. This is my current research group, and you've seen only a little bit of the fantastic work that they're doing. Previous students and postdocs are as many of them as I could find anyway. They've been scattered to the four winds, but they're some of them professors. They're working at companies. Startup companies, some of them are consultants, some of them work for government agencies, government labs, and so forth. And I'm very proud of the tremendous work they've done. Also, I would be doing practically nothing without the collaborations that I have. Almost all the work I do is in collaboration, and these are many of the collaborators that I've worked with. I want to especially mention Mike Solomon and Sharon Glotzer, with whom I've worked on a number of projects. Okay. I also need to thank the University of Michigan, which has a glorious history and a very bright future, I believe. Also, of course, I wouldn't be doing anything without the funding sources, so I want to thank them for supplying me the resources to be able to continue this work. And my family, very, very supportive. BB's here. I think I saw my daughter Rachel come in as well. Others are too far away. One's in China. Emily's in China right now teaching English. So she couldn't join me. And finally, I hope you'll indulge me for a moment. Because of my convictions, I feel I should thank God for all the good gifts that He gives me. And finally, I want to thank you, the audience, for coming to hear what I had to say. And thank you very much. I'd be glad to take questions from any of you.